Good afternoon, everyone. Let me introduce myself. I'm Jorge Galloso. I'm a hematologist. I'm the responsible of the programs for hematopoietic stem cell transplantation and cell therapy in the Organización Nacional de Trasplantes in Spain. And I will be the moderator for this second part of the immunotherapy session, which is devoted to the CAR T cells. We have a very good panel of speakers with us today. First of all, we have Manuel Juan. He's immunologist. He's the director of the immunotherapy section in the hospital clinic in Barcelona, and is responsible of the only academic clinical trial with CAR T cells in Spain. In the second part of the session, Manuel Martín Molina from Paco Martín Molina from the Genio, Spain, the Center for Research and Oncology in Granada. He will talk about the development of different kinds of CARs. Professor Locatelli finally will not join us today, and we will continue with Marta Duran. She's a pharmacologist, and she's the responsible of the development of the CAR program in Celgene in Spain, for Spain and Portugal in medical affairs, if I do correct. And finally, Enrique Granados from Gilead Science in Spain. Enrique is responsible for the development of the Gilead programs from all Europe. Thank you all for coming. We are interested in knowing about you. You know about us, and we want to make a very short survey to all of you. So please participate and raise your hands. Which countries you are from? Please, those who are from Spain, could you please raise your hands? Mostly Spanish. From another countries from the European Union? Welcome. And from other countries outside Europe? Well, okay. And what about your professional development? How could define you as a scientist or basic research? Or a clinician or pharmacologist? Okay. Or a representative or a member of a pharma company? So please don't be shy. And how many of you could consider yourself as investors interested in the field? So we have finished. If there is no money in the audience, so we are not very lucky today. So please, Manel, the floor is yours.
So I don't know, I don't move anything. Thank you very much for the introduction, and thank you for the organizer for inviting me to be here and to try to explain our short experience, because even we have been working for a long time ago in CAR T cells. I will just try to explain you some of our experience in the development of our academic CAR approach. I just put this first cartoon just to show you the basic elements that are involved in how the CAR is working. You need to have a target, and the tumor cell is the target for this approach. When we use CAR for cancer immunotherapy, because CAR could be used for other approaches, because finally you are using T cells and immune cells to try to target any kind of disease, but mainly in tumor cells. And in cancer, we use this as a target, and we have some kind of antigens. And when we are talking about CAR, we are using sometimes the final T to refer that the product that we use for treat these diseases are cytotoxic T cells, or general T cells, that introduce in some way a gene that codifies for a protein that it's in fact the CAR, the chimeric antigen receptor, the molecule that it's able to recognize the antigens in the tumor cells and by the recognition and after to be combined with several signaling domains, you can develop something inside these T cells, and for cytotoxic T cells, we are looking to have these several signals that induce cytolytic effect. But for have a good option to treat a real tumor with this approach is not enough to have this cytotoxic effect, that you need also to have other signals that make possible that this cell proliferate, make cytokines, and survive for at least enough time to kill all the tumor cells inside the body. This basic approach, or this basic idea where you are using these T cells, these modified T cells by a gene, and you use this cellular therapy with this modification of this molecule, has arrived to be successful when has been developed this approach by introducing the molecule that is called second generation CARs. Mainly the success arrived with when you use this CAR molecule, one recognize the CD19 molecule, because the main success arrived with leukemias and lymphoma, B-cell leukemias and lymphoma, who has this CD19 as a target. And after develop this molecule by molecular biology, you make finally the product that by changing the T cells. So to have this CAR product, we need to make at least, this is the more frequent CAR, we can use this as a current CAR. There are other development with 4th generation CARs and other approaches, but I will focus in this because it's also our approach for our proposal. But mainly you have to measure an element to produce the vector, or the way that you introduce this gene inside the cell, the product, and how to develop the cell therapy or the cell therapeutic product that comes mainly for autologous lymphocytes of the patients, that after this genetic manipulation by the vector, you need to produce in some way this vector or this way to manipulate. So it's possible also to make gene engineering, but mainly lentivirus has been used for, retrovirus has been used to modify these T cells. And after that, you need to grow these T cells. And probably it's also a major element to produce finally the enough quantity of T cells, modified T cells, to be used for reinfusion and to treat the patient with this product. This is probably the main scheme of how these CARs have been working. After several approaches and several companies and several clinical trials for CAR-19, the global results that I show or summarizing this quite old review article for more than 2 years ago, but the numbers doesn't change significantly for this. When you can see that for acute lymphoblastic leukemia, after for patients who has relapsed and refractory to any kind of treatment, you can obtain near a 90% of complete response, or a 90% of response, and finally a lot of these arrive to have complete response. Even after 1 year, the numbers decrease near a 50% of the patients. For non-Hodgkin lymphoma, the response is around the 40-50 also percent, but mainly of this response remains when they are complete, remains for a long time. For chronic lymphocytic leukemia, where the results seems were the first that has been focused in this target, but finally the result is at 25% of complete response. We need to improve a little to use this, but I think it's important to remember that this patient doesn't have good treatment for a long time. So this 25% of the patients could be also a good option for the patients. all these products for several companies, for several approaches, has arrived to be accepted for the clinic, and probably we are here talking about this because now they are already products for be used as a drugs, and these are the measures two products produced for two different companies, which is this is the unpronounceable name that is used for both, and one is mainly focused in the treatment of acute leukemia, but also some in the application of CD19 positive lymphomas. But the other is approved for the use in just one type of lymphoma. And here you have the prices accepted or proposed in states, in dollars. And the last quantification that we have comes for UK regulation that arrived near a little lower than this in the process on the price that finally could be applied for these products. I want to show you here some of our reflection about which are the more important things that it's necessary to have as access in an academic approach by using CARD. I think the more important thing, or at least the first, is to have a clear aim. And for us, and we think it's quite clear for us that it's not possible to have another final objective when we try to use the academic approach. Our aim is to achieve the hospital exemption, that it's a formal for authorization in just one hospital and just for some patients. This is for us the clear aim that we need to have. It's also too important to know how to arrive to obtain this clear hospital exemption, and by now the only way to arrive here is to make a clinical trial, as all the drugs that you want to obtain any kind of authorization. It's also important to have in mind how do you obtain the money to develop this clinical trial from an academic point of view. So you only have grants, and we know everyone here in Spain that it's very difficult to have enough money by grants to arrive to develop a clinical trial. It's possible to go to crowdfunding or another kind of funding to obtain this money. But finally, it's important to have success is to reduce everything, even the budget, to reduce also the risk of the proposal to obtain any kind of good results to arrive to be possible to develop this or to ask for this hospital exemption. And also very important, I think, if we want to develop this, is to be to organize everybody, to all the people necessary to develop this in a very multidisciplinary team. And it's very important the commitment of everybody in this. So if someone in your team doesn't want to move in this direction, I think it could be almost impossible to obtain the success in this option. Finally, we try to have all these concepts together in our approach, and we obtain the authorization of the Spanish agency to develop this clinical trial that we have here. And we develop our own card because we have for a long time ago some antibodies this kind against CD19 developed in our lab, and we use this as the receptor for this card, and we develop the structure of this card, and finally Spanish agency give us the opportunity to develop this clinical trial. The last week of May we begin to try to move and to treat these patients. And as a summary of all this data, now it's not possible to show you the results group in a more clear way because all this data should be evaluated by the Spanish agency. But here you can see all the patients that we have been treating by now. Each patient is a line. green line show that this patient has a complete response of the disease, the green one. When you have a red line, it's that the disease reappears again. It's a relapse of the disease. You have these two. One is CD19, this is CD19 negative. And when we have a black point or black line, it's the patient died finally for the relapse or for any other reason. It's not this moment because some of the patients have very few follow-up, and it's not the moment to analyze this. But in general, these are acute leukemia, these are lymphomas, and we have just one CLL. And we have similar results that has been described by now in all the other clinical trials. I want to remark the importance of the team to be able to coordinate all these people, more than 137 in our case, probably more than 150 by now, because each time we need something, we need other people who comes involved, and I think it's very important. But in any way, it's especially important to remark the role of the two clinical people who are involved, Dr. Delgado, Dr. Rivas, in the pediatrician and for the adult approaches. Also, the role that Dr. Ortiz is also making by managing all these things in the hematological point of view, and Miguel Caballero, who is developing the product in the immunology lab. Finally, just for make some comments about what it's made possible to develop this. I think it's very important, the concept that we are just looking a way to have this hospital exemption. We are unable to obtain all the money necessary to have a centralized approval, and then we think by this way will we be able to treat at least our patient and all the people who obtain, all the hospitals who obtain this hospital exemption by use this. I think this approach has as a main advantage is that the development of this academic can allow a faster evolution of better treatment. It's very difficult to develop a new treatment by new clinical trial in a centralized way in as fast as we need, or as fast we are able to develop scientific new concepts. So I think academical point of view is important. It's important the consistency in the process. It's very important that we are obtaining the beginning of all these things by public research grants, and after that we want to use this public health system, this research for public health systems. So in some way it's coherent that if we begin with money for the public research, finally this approach arrives here. And I think it's also important to have that by now, this CAR is not a universal CAR in the main approaches, and it's a very personalized way. And by using the French words, le boutique or la fabrique, I think we are these two options, and we are more near a boutique than a fabrique. So I think these autologous CAR are more near to be used as a personalized approach than to be used in a general, even probably in the following years, it will be the main source of the production of these CARs. So we put near the patient and the care. And so thank you for your time.
Okay. First of all, I would like to thank the organization for the invitation to give this brief talks in which I'm going to try to present what we are doing. We actually are vectorologists in Granada, so we are going to try to present what the CAR T cells from the gene therapy perspective, mainly how to manipulate the cells to actually try to do them a bit more efficient and or safer. Okay, so this is the initial scheme, which actually what you want to do with the CAR T cells is actually to have a T cells that you can have different ways to produce, to isolate, to expand, in which I'm not going to get into. And then you need to actually use, generally it's being used viral vectors to integrate the genes that codify for this chimeric antigen receptor that recognize the tissue antigen. So you have now a chimeric antigen that play the role of the TCR, recognizing the tissue-specific antigen and kill the tumor cells, okay? So for doing that, you need really to have the CAR gene integrated into the genome of the CAR T or the T cells, okay? And this is very important for the general strategy because the T cell is going to be dividing upon the antigen presentation, and each division is going to require that the cell express exactly the same amount of the CAR T cells of the CAR in order to have the same ability to destroy the tumor, all right? So this is very important. But you can also, there's a new strategy in which you can actually achieve that integration. You can use non-viral. And there's a new way in which you can do that by using transposons. By either of these ways, that actually you have good CAR technologies, and you actually have to control, and this at the moment has not been achieved. You need to control the expression of these CARs into the T cells, because if you have too high expression levels of the CAR, of this protein into the T cells, you have exhaustion of the T cell populations, and then the cells can go out from the patient before they kill all the tumor cells. But you can have also another problem, it's just CRS, which is the cytokine release syndrome, because the cell can be too much activated soon, and then you have a release of the cytokine storm, and that can kill the patients in some occasions. So you need actually to control that. However, if it's too low, the expression level you have here, then you don't have efficiency. And you are not able to kill the tumor cells. Then you have, again, the same problem if this regulation is not adequate. And this is the thing that in which our group, as a vectorologist or gene therapist, can actually do something. So you have a T cells in which you can actually have now there's other ways in which you can actually improve the efficiency, not only by the expression, but by using the genome edition tools that you hear before in the other sessions, to actually improve even further the ability of these T cells to be better. For instance, you can actually use the systems to knock down the TCR that is co-expressed with the CAR. So you have now the T cells that only express in the surface the chimeric antigen receptor. You don't have the endogenous TCR. And this is important by different reasons. One of them is actually you have the TCR chain can interact with the chain of the CAR and abrogate the expression level and have actually an expression on the surface that is not adequate. And one which is very important is that if you do this, you have a T cells that upon injection in the patient, you actually don't have graft-versus-host disease. So mainly by doing this strategy, you can have universal T cells that can be off the shelf and inoculate the patient as soon as they arrive into the clinic. So this is a quite nice and potent thing to do. But you can actually also do this, and you can eliminate immune checkpoints to actually try to do these T cells even more potent than they are by themselves. But if you do all of this, you really need to control the activity of the CAR T cells. Otherwise, you can have even higher severe side effects. So this needs to be controlled. So this is the aim of our lab. We started to work on this about 2 years ago now. So one of the things we wanted, because we actually do this in the lab, is actually to control the expression of the T cells, trying to mimic what actually King et al. Published in Nature, in which they published that if you control the expression of the CAR in the way that the TCR is expressed, then the CAR T cells are more potent, right? Because they actually do a TCR. This is what they do. A TCR does in the upon activation. You have 0 hours, just 12 hours upon encounter with the CD19 or either TCR antigen. What do you have actually down regulation of the TCR? Well, all the CAR expression systems that are at the moment in the market do the contrary. So you have this, and then the expression of CAR code here, because they use a different promoter. This is EF1 alpha or other viral promoters. So you actually, the actual system are quite badly designed this way, and this is the system at the moment in the clinic. The other thing that we want to do is actually try to control the activity of or the presence of the CAR into the T cells by the addition of doxycycline. And this is actually one of the things that we actually put a lot of effort in the lab. We have systems that we have patents that do not have transactivators, which are a problem for get this inducible system into the clinic. So we actually want to manage this in a system that is clinically plausible, right? You can actually go into the patient and put the system that you can add doxycycline to control the activity. And this is a system that actually we are working on a lot. And then finally, we have been working with the genome edition for 8 years now, and we have the tools to actually go into the different system, different T cells, knock down different genes, and either knock down the TCR or improve the efficacy by knocking down immune checkpoints. Okay, so we have all this technology. Let's do something of that. And what I'm going to show you is just small data we have in the lab, try to see the potency of the system. That try not to confuse about a lot of the data. So this is the T cell we have. We work a lot first trying to see which tools actually we use for genome engineering of the T cells. We managed to see that actually a system where we use the protein coupled to the guide RNA are the most potent. We managed to have about 80% of the knockdown of the TCR, which is quite enough to go further. We managed to design a Lenton Plus system that actually express the CAR T cells into an inducible way. That means that we have, at the end of this approach, we have knocked down the TCR, so the T cells do not express any endogenous TCR, do not express neither the CAR, unless we put doxycycline. If we put doxycycline, this cell come here, and the only thing that expresses is the CAR, the chimeric antigen receptor, right? So we can use a universal inducible system. This is just a panel of experiments to show you how we see this. And this is a dot plot, in which you can see about 80% of the knockdown of the TCR. This is the blue spots are T cells that do not express the TCR. And then we transduce the cell with the inducible system. Without dogs, you don't see here anything. And the purple colors are the cells that do express the CAR. Only upon addition of doxycycline, you see that. So we have about 20% of the cells that express inducible CAR upon the addition of doxycycline. So this is what I wanted to present you, and this is so what now? In which state are now? This is something. So this is for that. I'm going to talk about next time. This is something. This is a collaboration we have with Manuel Juan, in which we actually doing something on how to actually express these CAR molecules in a TCR way. I have some nice data. We are patented the system, collaborating with Manuel, so probably in the next meeting we have some presentation on this. In this system, actually, we really want to say that our inducible system really works on T cells without cloning or selection and without the use of transactivation. That is crucial for clinical translation. And we managed actually to have to say that in our lab, as well as in other labs around the world, it's really feasible to generate TCR knockdown that can generate inducible CAR T cells. So we actually want to join these two systems to generate inducible and universal CAR Ts and try to put that into the endothelial health system. And this is the people that had been involved in the lab. This general idea actually came through the IATA to me, because actually I was not even thinking about working on CAR T cells. But as a vectorologist, they just contacted me, and I thought it was a very good idea. Actually, it's working very well. I want to emphasize Karim Benadela, Maria, and Noelia as the key person in my lab to work on this. Of course, I have, from the very beginning, very nice contact with Hospital Reina Sofía with Concha Herrera, and Hospital Virgen de Rocío with José Antonio Pérez Simón. And nowadays, the University of Granada, through Juan Antonio Marchal, is helping me a lot with the animal models. And just to say thanks to the IATA and all the body fundings that helped me to carry on with this. Thank you very much.
Good afternoon. First of all, I would like to thank the organization to invite Celgene to this innovative roundtable about CAR Ts. Celgene is a biopharmaceutical company focused in the discovery, development, and commercialization of treatments for clinical unmet needs in cancer and immunoinflammatory diseases. Currently, Celgene have 30 drugs under development in different stages of development in hematology, targeting, proteostasis, epigenetics, and immunotherapy, with a novel special focus in cellular adaptive therapy with CAR-T. In multiple myeloma patients, there are 3 constructs under development, 2 in collaboration with Bluebird, BB2121 and BB2117, and 1 in collaboration with Immunopharmaceutics, GCAR125. And in patients with lymphoma and leukemia, we have another construct under development that is Lysogel, an anti-CD9 construct. BB2121 first data have been presented this year at ASCO and EHA, and I will summarize in the next slide. BB2121 is a second-generation CAR construct targeting BCMA, autologous T-cell transduced with a lentiviral vector encoding CAR-T, conjugated with co-stimulatory domain for 1BB, and T-cell activation domain CD3Z. Lysogel is an anti-CD9 construct. It's formulated at a specific composition of CD4 and CD8 T lymphocytes, and it's also autologous T-cell transduced with a lentiviral vector encoding CAR-T with a co-stimulatory domain for 1BB and T-cell activation domain CD3Z. So as I mentioned it before, preliminary data was presented at ASCO and EHA this year. This is a phase 1 dose-finding study in very pretreated patients with a median lines of 7 lines. And it was observed an overall response of 95.5% with complete responses of 50%. All patients evaluable for MRD negative were MRD negative, and comparable or overall response rates in patients with low and high BCMA expression. Dose response relationship observed across the active dose ranges. Higher PCAR-T expansion in responders versus non-responders, and median PFS was almost a year, 11.8 months in the global population, and almost a year and a half, 17 and 7 months in the responding patients. Regarding tolerability, adverse effects were manageable, mostly grade 1-2 CRS observed with infrequent tocilizumab and corticoid use. 2 events of grade 3 CRS resolved within 24 hours. No grade 4 CRS events were observed, and 1 case of reversible grade 4 neurotoxicity without additional events during expansion. Data in non-Hodgkin lymphoma patients was presented, the Transcend study, in 73 patients, and preliminary efficacy data show an overall response rate of 80% with complete response of 59%. Durable responses seeing 47% of patients with response at 6 months. Any CRS was observed in 37% of patients, with 1% more grade 3, and any neurologic effect in 25% of patients, more than grade 3 and 50%. So in conclusions, Celgene is building a preeminent global biopharmaceutical company focused in the discovery, development, and commercialization of innovative drugs for unmet clinical needs in cancer and immunoinflammatory diseases. For hematology, there are currently 30 drugs in different stages of development, targeting proteostasis, epigenetics, and immunotherapy with special focus in novel cellular adaptive therapy with chimeric antigen receptors, CAR-Ts. And preliminary results with CAR-T targeting B-cell modulation, antigen, and CD19 have shown deep and durable responses with a manageable safety profile to date in heavily pretreated population in multiple myeloma patients, and in poor prognosis patients in non-Hodgkin lymphoma, respectively. So these clinical programs are under development to confirm these encouraging results. Thank you.
Well, good afternoon to everyone. So thank you very much for inviting me to this session about CAR T cells. And my name is Enrique Granados. I'm working in Gilead since 4 years ago, but since the beginning of the current year, I am involved in the European implementation of the cell therapy in well, across from the medical department. So just my first slide is just to remember what the president of ASCO said this year at the opening of the American Society of Conference Oncologists, that the precision medicine is going to gain momentum across all the therapeutic armamentarium in the treatment of cancer. And even at the beginning of this year, ASCO point that CAR T is going to be one of the most important developments in the therapeutic space. And this has been followed by the decision of the European Commission a few weeks ago with the approval of the first 2 CARs in Europe and targeting different diseases in acute leukemia and diffuse great B-cell lymphoma. And well, it has been said already, but CAR T cells is a personalized engineering cells targeting using the patient own cells targeting different antigens. And in these 2 cases of the 2 CARs that has been approved, they are targeting CD19 that is expressed in normal and tumoral B cells, and this is the reason, the mechanism of action of these drugs. So basically, this is a quick summary of the pipeline of KITES. As you can imagine, once you get the approval in one disease, the next attempts are to try to treat patients at earlier stages. And this is being attempted, trying to target, to investigate with the same CAR in diffuse large B-cell lymphoma at second line, and using different approaches to increase efficacy and also to improve safety and increase efficacy by adding PD-L1 inhibitors. One of the features of this type of therapies is that from a regulatory point of view, modifications in the manufacturing process can result in being considered a different drug. And we are having 2 different manufacturing processes at this moment. One is the one that has been reached the approval by the EMA, but other are in research right now targeting diseases with leukemic expression of cells, targeting especially mantle cell lymphoma, pediatric acute lymphoblastic leukemia, and adult lymphoblastic leukemia. And as you can imagine, this special manufacturing mechanisms try to remove the circulating blast cells in blood. In addition to this CD19, we have another targets that are being investigated right now. For example, is BCMA that is expressed in multiple myeloma, and so it's another trial targeting multiple myeloma. And with regard to cell therapy, but not especially with CARs, so it is also the solid tumor space that it is really behind the situation in hematological malignancies. But some data, as a proof of concept, has been presented in last ASCO using antigens against the HPV, the human papilloma virus that you know quite well, that are related to head and neck cancer and different epithelial cancers. And we have made this with a good number of collaborations with different companies, and here is a summary of collaborations with companies or institutions as different as, well, the National Cancer Institute in the United States as a paradigm of private-public partnership. And we are very proud of this collaboration, and in fact it has been extended to study the neoantigens. You know, the neoantigens are tumors harbor a huge number of mutations. This huge number of mutations results in neoantigens that could be targeted by especially engineered T cells. And there has been a publication in breast cancer, in breast cancer with a very, very small number of patients. So this is just it must have viewed as just a proof of concept that it is promising. In addition to this collaboration with the NIH, we have some other collaborations with different gene editing techniques. In this case, it has been talked in the previous session about the CRISP, but we have an agreement with a company that are targeting or using the zinc finger nucleases to edit genes and to make these therapies with a better safety profile. As I said before, there are some also collaborations to increase efficacy by adding a PD-L1 inhibitor and also by adding a 4-1BB agonist. And one of the features of this technology is that it has a difficult or complex logistic process. This logistic process for sure will evolve in the future, and we hope that it will evolve once we have a manufacturing site in Europe, that it is expected by 2021. But for the time being, the logistic process involves collecting the cells of the patients, shipping them to the Netherlands, where they are frozen. From the Netherlands, they fly to the United States, where the transfection takes place, and the cells come back to Europe and make the release as a pharmaceutical product in the Netherlands, and from the Netherlands, they are shipped again to the initial hospital. And of course, there is a lot to do with regard to clinical research, but this is not enough if we need to bring these therapies to a reality in Europe and, of course, also in Spain. So we need to think how to implement this, and this is a number of open questions that probably we all have in mind. It involves the selection of sites. This selection of sites. The approach has been different from country to country. In the United States, it has been the decision of one private company putting in contact with another private company that there was a hospital. But across Europe, with the public health systems in place, the rules are really different, and so we are now awaiting the national, for example, in Spain, the national and the regional community authorities to have a say in the selection of these sites. The second point is how to train these sites. Dr. Manuel Juan talked before about the need to implement multidisciplinary teams, at least involving ICU specialists and neurologist specialists. I think that as a health system, there is the need to ensure a good equity in the patient access to these technologies. How to select the right patients is something that is going to be also important. And as a way to summarize, we are here to collaborate with hospitals and health systems to try to implement this technology as soon as possible. And I think that's all for my side.
Yeah, okay, sorry. congratulations for these talks. They were very good, all of them. This is a question for the whole panel. As you've been presenting, and as it looks like, both the academic and the commercial CAR-T will come pretty much at the same time in the hospitals. So I would like your opinion on how this coexistence will be managed in terms not only from the clinical perspective, but also from the academic, logistics, and budget also, because we'll have the academic CAR-T, which probably with less price and maybe with advantages in the logistics, but also we'll have, on the other hand, the commercial with larger trials and also with more safety studies behind them. So how do you think this will coexist?
I don't know exactly how will we coexist, but I think it's possible, the coexistence, because finally it's almost impossible that from the academic point of view it's possible to arrive to everybody, in sometimes too many patients. But probably finally the more limiting aspect will be the development of this multidisciplinary team in each center. I think even everybody wants to be involved in this because the patients are in all the places. Finally, it will be very important to define where this treatment will be used and will be produced and infused to the patients, and to define the centers and how these centers will be managed this problem, I think will be the first step that authorities should decide.
Well, if I can add something, probably my answer would be I do not have the full picture at all. So it's probably impossible to say for none of us to foresee how this could be in 5 years, is how we are going to coexist. But our view is that there is room for both. It is clear that the academic centers are doing excellent clinical research, and there is a room for growing the knowledge of this area. There is an incredible room for increased knowledge, but I think that at the same time, probably the commercial cards are the best way to ensure that once in some indication has been reviewed by the health authorities and ensuring all the process of GMP and review of the dossiers and so on.
Sorry, I would like just to comment that for academic CAR-T cells, the requirements are exactly the same. So they have to comply with GMP regulation, and for hospital exemption, at least in Spain, not in other countries, but at least in Spain, the requirements are the same than for a central commercialization, marketing authorization. They have to comply with a CTD, and if you are not able to provide enough information related to preclinical evidence and clinical evidence and quality evidence, you are not going to be granted hospital exemption.
That's it. It's the only way to arrive, but I think the rules are for everybody, and I thought that if we have these rules, it will be possible to arrive to obtain this level of quality.
I have also a question for both positions. Somebody from the academic and somebody from the pharmaceutical companies. Is there really any evidence of the cost effectiveness of these treatments right now? With the data we have, how could a regulatory office proceed in the order to authorize the use of these drugs in our country, and how should we deal with so high prices and these actual results. Most patients get responses, but they are not durable. Less than 0.5% of the patients lose the response at 6 months or so, in leukemia also, and in lymphoma. So should we pay 300,000 euros per treatment?
Well, I think it's the price is still something that is not now. So but you know, it's quite when you compare with the current situation, or with the current results that these patients have. I think that, and even acknowledging that the trials are single arm, most of them. So at least what has been approved by the European Commission, both are single arms, so there's no control arms with not a huge number of patients. I think that the results are clear better than the current therapies. So I'm quite sure that the cost effectiveness will be part of the discussions with the authorities in terms of the reimbursement, and probably. There will be need to consider other features, like the fact that this is a single shot treatment, and it makes some difference, or clear difference from other classical drugs that needs a chronic administration, and how at the end this is managed let's say from a reimbursement perspective. I think this is something that we need to find the way.
Well, no, I have not much to add, but of course we need a longer follow-up, and I think that governmental affairs will need to find ways to discuss the authorities and the pharmaceutical companies to find a way to at the end show to have this available for all the patients.
But at the moment, the only evaluation that I know is that from NICE, from the UK, and they have not approved the reimbursement of your card, and also they have not approved the reimbursement of the card for lymphoma from Novartis. They only have approved the reimbursement of the card for acute lymphoblastic leukemia in a small number of children. So there are also other opinions from the States, from independent institutions, that CAR T-cell therapy is not always cost-effective. So what's the situation?
Well, so the response from the NICE is not unexpected at all. So I think this is, I think this has been the decision of NICE with most of oncology products in the last 10 years. And probably there are some features of very different, in this case, that in the NICE, like as I said before, the single shot. To the best of my knowledge, in UK, they are now trying to get approval through the Cancer Drug Fund. That it is not exactly the same as NICE. I'm not completely familiar with this, but I think they are open to treat it with the Cancer Drug Fund. The fact that it has been approved in pediatric indication, well, this came to the clear, I think, acknowledgement by all of us of the value of the drugs. It's a pediatric indication, and the time cost life saving probably is better. And United States, you made a question about United States. So I think that the situation of the health systems between United States and Europe are so different that it is very completely impossible to extrapolate the resources needed in a hospital in the United States to the resources needed in a hospital in Europe, and in terms of costs. And in terms of integrated payer and not multiple payers like they are in the United States. So I think they will need to go to Europe and to see how the different health systems can accommodate this.
So it's very good debate. My name is Massimo Domenici from Italy. I'm actually pointing out on the type of collaboration that might be needed between public and private provision of this CAR-T, because there is no doubt that the companies are investing in this field, and there's no doubt that public institutions will do the same. So maybe, we should envisage a partnership amongst the two players, because even in the public structure, there will be a cost for manufacturing of the cells, which will not be trivial. It's going to be a cost that might not be as exactly the same as for the companies. So maybe a system to compensate those costs of public costs in collaboration with companies is a business model that could be explored. It's something which is pretty unique. But I really feel the need that we need to explore that at the academic level, at the hospital level. So the cost is 300,000. Maybe the part of this cost can be used for support public production of CAR-T cells. It's a bit provocative, but it's something that we need to think about.
I don't know if it's possible, at least here in Spain. Probably in other countries, the collaboration are easier than here, but we have been exploring this collaboration for more than 4 years at the beginning, and it was almost impossible. I think major companies are involved in this approach, and they decide how they manage, and they doesn't want that nobody in the academics define other aspects. So it's quite difficult to arrive this collaboration in any kind. I think it could be fine. And probably one of the aspects that it could be easier is to make possible the production next to the patient, because finally it's a procedure and it's possible to develop, but most of the company wants to centralize this production, and maybe this centralization make a little more complicated the collaboration. I don't know. I think it's absolutely open to any kind of options, but maybe are easier the collaboration in the States and even in some biggest countries in Europe, but not at least in Spain.
Sorry for intervening again, but well, maybe the maximum comments is not only referred to a public-private collaboration in terms of the development of CAR-T cells. Maybe what he, I guess, or at least is my opinion. So then, what we think is that the efficacy of these treatments, it doesn't depend exclusively of the quality of the medicinal product. So then the results of these therapies depends on the collaboration of the healthcare system with companies. So in some way, quite clear that CAR-T cells are maybe one of the advanced therapy medicinal products more similar to transplants. So then we can identify 3 different steps in this treatment. It's like a procedure. There is a procedure of donation, there is a procedure of manufacturing, and there is a procedure of delivery. So then, of course, companies have an important role in manufacturing, in making possible the manufacturing for a lot of population that from academic centers is very difficult just to be possible to treat all patients or all the necessities. But companies need someone extracting the lymphocytes to send the lymphocytes to the center to be manufactured, so then to send the CAR-T cells to the hospitals, so then to organize all the logistics in order to deliver the treatments. We need multidisciplinary teams in hospitals. We have well-trained professionals at those hospitals, so then the opportunities for collaboration are incredible from companies and the public healthcare system. That is maybe what we need to think about, how to pricing this kind of medicinal products in which what the company is really doing is providing a service. That is my point of view.
Thank you so much. There is no other questions or comments from the audience. Thank you very much for attending this session, and we finish here. Thank you so much.